friends, today I am going to discuss the topic general mechanism of immune system. The main objectives of this lecture are to understand the basic concept of immune system, innate and adaptive immunity, phagocytosis and inflammation, different cells involved in immune system. First of all, introduction. The immune system is a remarkably versatile defense system that has evolved to protect animals from invading pathogenic microorganisms and cancer. It is able to generate an enormous variety of cells and molecules capable of specifically recognizing and eliminating an apparently limitless variety of foreign invaders. These cells and molecules act together in a dynamic network whose complexity rivals that of the nervous system. Functionally, an immune response can be divided into two related activities. First, recognition and response. Immune recognition is remarkable for its specificity. The immune system is able to recognize subtle chemical differences that distinguish the foreign pathogen from another. Furthermore, the system is able to discriminate between foreign molecules and the body's own cells. Now, the historical background. The discipline of immunology grew out of the observation that individuals who had recovered from certain infectious diseases were thereafter protected from the disease. The Latin term immunus, meaning exempt, is the source of the English word immunity, meaning the state of protection from the infectious disease. Edward Jenner in 1798 intrigued the fact that milkmaids who had contracted the mild disease cowpox were subsequently immune to smallpox. Jenner reasoned that introducing fluid from a cowpox pustule into the people might protect them from smallpox. To test this idea, he inoculated an 8-year-old boy with fluid from a cowpox pustule and later intentionally infected the child with smallpox. As predicted, the child did not develop smallpox. Now, the immune system includes innate and adaptive components. Immunity has both a less specific and more specific component. The less specific component, innate immunity, provides the first line of defense against infection. In contrast to the broad reactivity of the innate immune system, which is uniform in all members of a species, the specific component, adaptive immunity, does not come into play until there is an antigenic challenge to the organism. Now, first of all, innate or non-specific immunity. Innate immunity is present from birth and consists of many factors that are relatively non-specific. That is, they operate against almost any substance that threatens the body. It resists infection by blocking the entry of pathogens into the body or by destroying the microbes through means other than antibodies. One form of non-specific resistance is called species immunity. Innate immunity comprises 4 types of defense barriers: anatomic, physiologic, phagostic, and inflammatory. Now, first of all, external defenses, or we can say first line of defense. This defense comprises physical and chemical barriers to the entry of pathogens into the body. First of all, physical barriers. These in turn are of 2 kinds: skin and mucous membranes. Let's talk about skin first. Skin provides a protective covering to the body. Its outer tough layer, called horny layer or stratum corneum, consists of dead, fully keratinized cells. These cells contain a hard, insoluble fibrous protein called keratin. This layer is waterproof and germ-proof, thus preventing the entry of viruses and bacteria. Now, mucous membranes. The digestive, respiratory, and urinogenital tracts open out at one or both the ends and do not have a direct communication with other parts of the body. The parasites present in these tracts are not in the physiological interior of the body. The mucous membranes lining these tracts are therefore treated as a part of the external defense, like the skin. The mucous membranes resist the penetration of parasites into the tissues. Mucus secreted by mucous membranes trap the microorganisms and immobilize them. Now, eyes. Secretion of tears and movement of eyelids flush out the microorganisms settling on the eyeballs from the air. Now the chemical barriers. The skin and the mucous membranes secrete certain chemicals which dispose of pathogens, skin secretions and bacteria. The oil and sweat secreted by the sebaceous and the sudoriferous glands contain fatty acids and lactic acid, which make the surface acidic. This does not allow the microorganisms to establish on the skin. Now saliva. Saliva also contains lysozyme, which kills the microbes that are not the normal inhabitants of the buccal cavity and comes with the food and drinks. The dead microbes are then passively flushed by the saliva to the throat where they are swallowed. Now the gut secretions and bacteria. The microbes may escape the action of the saliva and reach the stomach. Bacteria trapped in the mucus in the respiratory tract also reach the stomach when swallowed. Here they are killed by hydrochloric acid and proteolytic enzymes of the gastric juice. The proteolytic enzymes in the small intestine also help in killing the microorganisms. If the bacteria survive and reach the large intestine, they are attacked by the gut microorganisms which secrete antibiotics that kill many pathogenic bacteria. Now bile. Bile is a bitter alkaline secretion of the liver which checks the growth of the foreign bacteria on the semi-digested food. Tears. Tears, a slightly saline fluid secreted by the lacrimal glands over the eyes, also contain lysozyme which prevent eye infections. Now we have got the nasal secretions. Nasal secretions also destroy the harmful foreign germs with their lysozyme. Now cerumen. Cerumen or earwax, a bitter brownish secretion from the ceruminous glands in the auditory canal, traps dust and bacteria. It contains an effective antibacterial component and also prevents entry of insects. Now we will talk about the penetration of the first line of defense. Skin and mucous membranes may fail to keep out the invaders. Some parasites make way through the skin, example hookworm. Others enter through the wounds and openings of the sweat glands and hair follicles. Still others are injected by the blood-sucking arthropods. Microorganisms may injure and pass through the thin, moist, relatively vulnerable mucous membranes of digestive, respiratory and urinogenital tracts and get into the tissues and blood. It is therefore very essential for survival to have a second line of defense for controlling the parasites. Now the second line of defense comprises of internal or cellular defenses which include physiologic, phagocytic and inflammatory response. First of all, physiologic barriers. Physiologic barriers to infection include general conditions and specific molecules. The physiologic barriers that contribute to innate immunity include temperature, pH, and various soluble and cell-associated molecules. Many species are not susceptible to certain diseases simply because their normal body temperature inhibits growth of pathogens. Gastric acidity is an innate physiologic barrier to infection because very few ingested microorganisms can survive the low pH of the stomach contents. A variety of soluble factors that contribute to innate immunity are soluble protein, lysozymes, interferon, and complement. Lysozyme, a hydrolytic enzyme found in mucous secretions and in tears, is able to cleave the peptidoglycan layer of the bacterial cell wall. Interferon comprises a group of proteins produced by virus-infected cells. Among the many functions of the interferons is the ability to bind to the nearby cells and induce a generalized antiviral state. Complement is a group of serum proteins that circulate in an inactive state. A variety of specific and non-specific immunologic mechanisms can convert the inactive forms of complement proteins into an active state with the ability to damage the membranes of pathogenic organisms. Complement may function as an effectory system that is triggered by binding of antibodies to certain cell surfaces, or it may be activated by reactions between complement molecules and certain components of microbial cell walls. Now let's talk about the cells of the immune system. Cells that ingest and destroy pathogens make up a phagocytic barrier to the infection. Another important innate defense mechanism is the ingestion of extracellular particulate material by phagocytosis. Phagocytosis is considered a major form of non-specific defense in the body. The cells involved are called phagocytes. Most phagocytosis is conducted by specialized cells such as blood monocytes, neutrophils, and tissue macrophages. Now, neutrophils. Neutrophils are the most abundant phagocytic cells in the blood. These are also called as polymorphonuclear leukocytes. They respond to chemotactic signals and leave capillaries by a complex process involving margination, rolling, and then attaching, following which they emigrate between the endothelial cells. Now the basophils. Basophils are non-phagocytic cells which when activated release numerous compounds from the basophilic granules within their cytoplasm. They play a major role in allergic responses. Now monocytes. Monocytes circulate in the peripheral blood prior to the emigration into the tissues. Within certain organs, they have special names. Example, in liver, they are known as Kupffer cells, in brain as microglia, and in bone as osteoclasts. Elsewhere, they are referred to as tissue macrophages. Now, let's talk about these macrophages. These cells derive from bone marrow promonocytes which after differentiation to blood monocytes finally settle in the tissues as mature macrophages where they constitute the mononuclear phagocytic system. They are present throughout the connective tissue and around the basement membrane of small blood vessels and are particularly concentrated in lungs, liver, and lining of spleen, sinusoids, and lymph nodes, medullary sinuses, where they are strategically placed to filter off the foreign material. Macrophages are at their best in combating those bacteria, viruses, and protozoa which are capable of living within the cells of the host. Macrophages are actively motile, adherent, and intensely phagocytic. In addition, they can be activated by lymphokines. Now, what are the steps involved in phagocytosis? Mononuclear phagocytes exhibit non-immune and immune phagocytosis. This process consists of several steps. First of all, recognition and attachment to the particle to be phagocytosed, actual engulfment, killing and digestion, and post-digestion, that is disposal of the remnants. During phagocytosis, foreign particles are bound to either specific or non-specific receptors and then surrounded by the cell membrane to form a phagocytic vesicle. Alternately, soluble macromolecules might be pinocytosed in a similar fashion. FCRs and CRs enable the macrophages to recognize particles coated or opsonized with antibody and complement molecules. However, some antibodies are cytophilic. These antibodies bind to the macrophage first, then bind to the antigen or the particle to be phagocytosed. In this manner, immunological specificity for the target particles is acquired. Bound soluble and insoluble materials are then ingested. Following phagocytosis, formerly interiorized receptors are re-expressed on the macrophage membranes. Engulfment is accomplished by physical wrapping of the membrane around the particle. This creates a phagocytic vesicle or phagosome which is taken into the cell. Once formed, the phagosomes are moved within by microtubules. They then fuse with lysosomes forming phagolysosomes. Next, the contents become acidified and are digested by a host of hydrolytic enzymes. Now let's talk about another reaction which is inflammation. Inflammation is the complex biological response of vesicular tissues to harmful stimuli such as pathogens, damaged cells, or irritants. It is a protective attempt by the organism to remove the injurious stimuli as well as initiate the healing process for tissue. In the absence of inflammation, wounds and infections would never heal, and progressive destruction of the tissue would compromise the survival of the organism. However, inflammation which runs unchecked can also lead to diseases such as hay fever, atherosclerosis, and rheumatoid arthritis. Inflammation is characterized by four cardinal signs, that is, rubor, tumor, calor, and dolor. The cardinal signs of the inflammation reflect the three major events of an inflammatory response, which are as under. First of all, vasodilation, which is an increase in the diameter of the blood vessels resulting in the engorgement of the capillary network. The engorged capillaries are responsible for tissue redness and an increase in the tissue temperature. Another, an increase in the capillary permeability facilitates an influx of fluid and cells from the engorged capillaries into the tissue. The fluid that accumulates has much higher protein content, and its accumulation contributes to the tissue swelling. Another is influx of phagocytes from the capillaries into the tissues is facilitated by the increased permeability of the capillaries. The immigration of the phagocytes is a multi-step process that includes adherence of the cells to the endothelial wall of the blood vessels, followed by their emigration between the capillary endothelial cells into the tissue, and finally their migration through the tissue to the site of invasion. As phagocytic cells accumulate at the site and begin to phagocytose bacteria, they release lytic enzymes which can damage nearby healthy cells. The accumulation of dead cells, digested material, and fluid forms a substance called as pus. Now let's talk about the adaptive immunity. Adaptive immunity, in contrast to the innate immunity, which is an attribute of every living organism, acquired immunity is a more specialized form of immunity and always exhibits specificity. It developed late in the evolution and is found only in vertebrates. As its name implies, acquired immunity is a consequence of an encounter with a foreign substance which, when penetrates the body, triggers a chain of events that induces an immune response with specificity against that foreign substance. There are two different forms of the immune system. One, antibody mediated immunity, which is also called as humoral immunity, in which specific proteins, the antibodies, are made when foreign antigens are detected. An antigen is any macromolecule that elicits the formation of an antibody and that can subsequently react with an antibody. In this kind of response, plasma cells derived from certain white blood cells, B lymphocytes, synthesize antibodies in response to the detection of a foreign macromolecule with antigenic properties. Antibodies, also called as immunoglobulins, are made in response to specific antigens and react with those antigens. Now let's talk about the cell-mediated immunity, which is also called as cellular immunity, in which certain cells of the bodies acquire the ability to destroy other cells that are recognized as foreign or abnormal. This type of response depends on another class of lymphocytes or cells which we call as T lymphocytes. These cells interact with the foreign cells to destruct them. There are also interactions between B and T lymphocytes and between these and other blood cells that establish an integrated defense network. Now let's talk about the types of immunity. Specific or adaptive immunity, which is often subdivided into two major types depending on how the immunity was introduced. Natural immunity occurs through contact with a disease causing agent when the contact was not deliberate, whereas artificial immunity develops only through deliberate actions. Both natural and artificial immunity can be further subdivided depending on the amount of time the protection lasts. Passive immunity is short-lived and usually lasts only a few months, whereas protection via active immunity lasts much longer and is sometimes lifelong. Now let's talk about the active immunity. This kind of immunity occurs as a result of infection. When exposed to an infectious agent, the body starts manufacturing its own antibodies. On exposure to first infection, memory cells are produced, leading to immunological memory. Due to the formation of immunological memory, reinfection at later time leads to a rapid increase in antibody production and effector T cell activity. When B cells and T cells are activated by a pathogen, memory B cells and T cells develop. Throughout the lifetime of an animal, these memory cells will remember each specific pathogen encountered and are able to mount a strong response if the pathogen is detected again. This type of immunity is both active and adaptive because of the body's immune system prepares itself for the future challenge. Active immunity often involves both the cell-mediated and the humoral aspects of immunity as well as input from the innate immune system. The innate system is present from birth and protects an individual from pathogens regardless of experiences, whereas adaptive immunity arises only after an infection or immunization and hence is acquired during life. Now let's talk about the naturally acquired active immunity. Naturally acquired active immunity occurs when a person is exposed to a live pathogen and develops a primary immune response which leads to immunological memory. This type of immunity is natural because it is not induced by man. Many disorders of immune system function can affect the formation of active immunity such as immunodeficiency disease and immunosuppression. Artificially acquired active immunity. Artificially acquired active immunity can be induced by a vaccine, a substance that contains antigen. A vaccine stimulates primary response against the antigen without causing symptoms of the disease. The term vaccination was coined by Edward Jenner and adapted by Louis Pasteur for his pioneering work in vaccination. The method Pasteur used entailed treating the infectious agents for those diseases so they lost the ability to cause serious diseases. Various types of vaccines used are inactivated vaccines, which are composed of microorganisms that have been killed with chemicals or heat and are no longer infectious. Examples are vaccines against flu, cholera, and hepatitis. Most vaccines of this type are likely to require booster shots. Live attenuated vaccines are composed of microorganisms that have been cultivated under conditions which disable their ability to induce disease. These responses are more durable and do not generally require the booster shots. Examples include yellow fever, measles, and mumps. Another, toxides or inactivated toxic components from the microorganisms used prior to an encounter with a toxide. Examples of toxide-based vaccines include tetanus and diphtheria. Another vaccine which we have got is a subunit vaccine, which is composed of small fragments of disease-causing organisms. A characteristic example is the subunit vaccine against hepatitis B virus. Most vaccines are given by hypodermic injection as they are not absorbed reliably through the gut. Live attenuated polio and some typhoid and cholera vaccines are given orally. Now, let's talk about the passive immunity. Passive immunity is the transfer of active immunity in the form of ready-made antibodies from one individual to another. Passive immunity can occur naturally when maternal antibodies are transferred to the fetus through the placenta and can also be induced artificially when high levels of human antibodies specific for a pathogen or toxin are transferred to the non-immune individuals. Passive immunization is used when there is a high risk of infection and insufficient time for the body to develop its own immune response, or to reduce the symptoms of ongoing or immunosuppressive diseases. Passive immunity provides immediate protection and the body does not develop memory. Therefore, the patient is at the high risk of being infected by the same pathogen later. Now, naturally acquired passive immunity. Maternal passive immunity is a type of naturally acquired passive immunity and refers to antibody mediated immunity conveyed to a fetus by its mother during pregnancy. Maternal antibodies are passed through the placenta to the fetus. This occurs around the third month of gestation. Usually, IgG is the only antibody isotype that can pass through the placenta. Passive immunity is also provided through the transfer of immunoglobulin A antibodies found in the breast milk that are transferred to the gut of the infant, protecting against bacterial infections until the newborn can synthesize its own antibodies. Now, let's talk about the artificially acquired passive immunity. Artificially acquired passive immunity is a short-term immunization induced by the transfer of antibodies which can be administered in several forms as human or animal plasma or serum, as pooled human immunoglobulin for intravenous or intramuscular use, and in the form of monoclonal antibodies. Passive transfer is used prophylactically in the case of immunodeficiency diseases. It is also used in the treatment of several types of acute infection and to treat poisoning. Immunity derived from the passive immunization lasts for only a short period of time, and there is also a potential risk for hypersensitivity reactions and serum sickness, especially from the gamma globulin of the non-human origin. The artificial induction of passive immunity has been used for over a century to treat infectious diseases, and prior to the advent of the antibiotics was often the only specific treatment for certain infectious disease. This was all about immune system. Hope you would have understood it well. Thank you very much.